So our first speaker today is Oracle Banerjee. He's currently a postdoc fellow at NYU Medical Center with Michael Long. But he is starting as assistant professor at Cold Spring Harbor Lab from next Monday. So congratulations. And Oracle is interested in neurocircuit mechanism of ethologically relevant behaviors. For his PhD, he studied neurocircuits for sensory processing in a factory system. And as a postdoc, he worked with an exotic rodent species, colloquially called the singing mouse. And he currently holds the world record for recording every action potential has ever been recorded from this species. So Oracle, the floor is all yours.
Thank you very much for the kind introduction. I'm extremely excited to be sharing with you our adventures with the Alston singing mouse. They're native to cloud forests of Costa Rica. As a systems neuroscientist, I am interested in understanding how sensory information is processed by the brain to generate appropriate motor outputs. And typically, one way to study these in the lab would be to train an animal, for example, a rodent here, to associate a sensory stimulus with a motor action, for example, from the Saboda lab, where the pole position is associated with the direction in which the animal has to lick to indicate his choice. But we are in a session about vocal communication, and I would like to make the point that vocal communication is ideally suited to study sensory motor transformations. Because, for example, in conversation, you have to process the auditory information, and then after a stage of planning or cognition, the motor cortex or the motor system in general swings into action and you have to produce the motor output. And this can happen in a very dynamic setting and can go back and forth. And what's really exciting and interesting is that it takes roughly about 200 milliseconds, which is remarkably small. Now, then the question becomes that how can we study such dynamic sensory motor transformations in the lab? And this is where I get to introduce to you the protagonist of today's story, the Alston singing mouse. They are highly vocal rodents from Costa Rica and Panama, and they're remarkable in their ability to not only sing a stereotyped song that lasts over tens of seconds, but also in their ability to go back and forth, and that's how they communicate. So what I'm going to play to you now is a video that I recorded in Michael Long's lab at NYU, where you will hear the mouse on the left sing a full advertisement song and then a response from the mouse on the right. And mind you, they can't see each other or smell each other, but they can hear each other. Okay, here we go. All right, so now these mice can participate in this dynamic vocal exchanges hundreds of times a day. Here is just 1 hour of continuous recording from these two mice, mouse 1 and mouse 2. And you can see that they actually sing fairly good amount of songs over 1 hour. But if you look closely, you will realize that they're not singing randomly. So for example, here you see that mouse 1 sings a full song that evolves over maybe 7 seconds, followed by a response from mouse 2, which I'm showing in red here. And if you do the statistics and look across many such interactions over many pairs of animals, what you see is that typical latency is around 500 milliseconds, which is quite fast. Now, one hallmark of social behavior is their ability to change motor output depending upon what the conspecific animal is doing. So in order to show you that, I need to first describe to you how we can represent one song. So a typical song is a series of notes that evolve over tens of seconds with a very stereotyped progression, which is that notes that happen at the beginning of the song are shorter and they become progressively longer as the song progresses. And you can plot this as this trajectory plot where note duration increases as a function of time. And what I'm showing here on the right is that if you take 15 songs on a random basis from this animal that's singing solo songs just on its own, they're remarkably stereotyped. They are all on top of each other. However, the moment they can hear another animal and starts participating in vocal exchanges, that stereotypy is broken And you can see that the song becomes shorter, even longer, and the rate at which the notes increase or decrease is variable. So I have shown you so far that the singing mice can participate in very fast vocal exchanges, and they can also modify their motor output based on what they're hearing. Okay, so with this background, I want to switch gears and try to understand what are the neural circuits that underlie such vocal communication in the species. Okay, we need to begin somewhere. And Daniel Okobi, who was a grad student in Michael's lab before I joined, he did a very interesting experiment where he electrically stimulated large areas of the dorsal cortex, trying to record from EMG activity in the vocal musculature, and found that an anterolateral portion of the motor cortex, when stimulated, results in a robust flexion of vocal muscles. So we call this area orofacial motor cortex, or OMC, because it overlaps with the generic orofacial motor cortical areas in other rodents as well. Okay, so then one can wonder, what's the role of OMC in this behavior, which is called counter singing, this ability to go back and forth? So I pretended to be mouse one in this case and played back songs from a speaker, and mouse two was adequately fooled into responding very robustly. So you can see that mouse two songs are in red and they're responding quite robustly to the playback. But if I inject mucimol and inactivate OMC, their ability to respond to playback falls apart. And even in cases where they manage to respond, it has a much longer latency. I can quantify this across many animals, and it was quite a robust effect where the ability to respond back to playback was significantly hampered if OMC was inactivated with mucimol. I also noticed something else, which is that if I quantified the duration of the songs that were produced in either saline or mucimol, you could see a shift such that the song durations after inactivation were comparatively shorter. Okay, so these two observations, this ability to participate in sensorimotor transformation, which got hampered, as well as the song duration distribution that got lowered, led us to make some predictions. So the predictions are pretty simple. One is that we should expect that there should be neurons in OMC that would be differentially modulated between solo songs and counter singing, if this area is actually important in counter singing. And second, that there would be neurons here in OMC whose activity should co-vary with song duration, because inactivating OMC changed the song duration. And in order to test these predictions, I collaborated with a grad student in the lab, Margot, to develop a recording system in which, with chronic electrophysiology with high density silicon probes, I could record motor cortical neurons for weeks, maybe up to a month. And what did we find? Here is prediction number one. This is a neuron, and I'm showing you 30 trials where this animal, mouse 2, was implanted and he sang 30 songs. And this is a raster plot. And you see this neuron's firing rate tends to peak right about when mouse 2 starts singing. However, we can do a trick here. Not all trials are the same. Some trials are when the mouse sings spontaneously, so that those would be motor trials. There's some trials when the mouse responds to a song that I've played back, so those would be sensory plus motor trials. And then there are trials where I have played back a song, but the mouse did not care to respond, so those would be sensory only trials. And if we split the activity of this same neuron into these three contexts, I think you can see that they show context dependence. For example, during the solo condition, the neuron, again, fires just around the animal's start of song. However, during counter singing, when the animal is listening, the activity starts departing from the baseline around 10 seconds before the animal has to sing, which is this red line here. And now if you think that that reflects the auditory activity, it doesn't seem to be so, because the neuron does not seem to care about pure auditory stimulus, which is the green line here, okay? So it seems that neural activity in this region, and giving you an example, but it's true at the population level as well, is predictive of the decision to counter sing. How about the second prediction? Well, the second prediction was that there should be neurons here whose activity should co-vary with song duration. And again, here I'm showing you another neuron I'm aligning the activity of this neuron to the beginning of the mouse's song, so that song start, and then I'm sorting all the trials such that the shortest song is on the top and the longest song is at the bottom. And perhaps you can see that the activity of this neuron tends to monotonically vary with the duration of the song. And let's make it more explicit. If you just make a PSTH of this neuron for songs of different durations, what you should see is that the PSTH seems to systematically vary with respect to the song of the overall song duration. Moreover, what really seems to be happening is that the short songs PSTH, when artificially stretched to match the song duration of the long song, seems to be very similar, right? So we have done quantifications to show this in a more quantitative regime, but the upshot is that the long song's neural activity seems to be a stretched version of the short song, okay? So then one can ask the following question, and okay, there seems to be some neural dynamics that can track this overall song duration, but how do you know if this neural activity is causal for behavior? We can address this question by actually physically cooling down OMC. And the idea is as follows: if you cool down OMC, the neural dynamics slows down. And if neural dynamics in this region has anything to do with the behavior, you should observe a concomitant slowing down of the behavior. And that's exactly what we see. So this is on the right, control song at body temperature, and it evolves around, takes around 7 seconds, but progressively with lowered temperatures, the song elongates in time, which actually shows that neural activity in OMC that compresses and stretches to match the dynamics of the overall song actually can determine song duration causally. Okay, so with that, I would like to conclude by saying that what I have shown you today is the behavior of the singing mouse and how they can participate in vocal exchanges with each other at a very fast time scale, and also their ability to modify their vocal output based on what they're hearing. So it's a legitimate sensorimotor transformation. And through various perturbation experiments and electrophysiological recordings, I hope that I have convinced you that motor cortex is required for vocal interactions in this rodent. And there are socially modulated neurons in motor cortex that distinguish between solo versus duet songs. Moreover, neural activity in this brain region seems to determine overall song duration. And what all of these tend to suggest is that there is robust motor cortical control over vocalization in a rodent, and it's not restricted to the primate lineage. And before I end, I would like to sort of advertise how interesting the singing mice species is as a model system, where you can ask fundamental systems neuroscience questions of how you can go from behavior to physiology and manipulations using viruses in a sensorimotor transformation, but then also ask evolutionary questions of what changes might need in neural circuits that allows for different behavior to evolve along similarly related species. So for example, on the top here is mus musculus USVs, and we have 2 fantastic talks lined up that will explore that. So stay tuned. And in closing, I would like to acknowledge Michael Long, my postdoctoral advisor, for allowing me to work on this exciting project and for his critical insights over these last many years. And none of this would be possible without Steve Phelps, who is a long-term collaborator who pioneered this singing mouse model system. And if anybody in the audience is interested in pursuing some of these questions as a student or a postdoc at Cold Spring Harbor Labs, please feel free to either email me or contact me over Twitter. Thank you very much.
Thank you very much for a very interesting talk. We have many questions, so I will read them. And the first one from Brandon is, are these mice lifelong vocal learners like budgerigers? I'm sorry, I don't know how to pronounce that. Or is it crystallized on more similar to zebra-finches? If they are vocal learners, any idea if OMC affects song and response quality or learning?
That's a great question. what we know so far is that their ability to produce these stereotype songs seems to be innate. And there's some experiments that have been done by Steve Phelps that suggest that. What is not known is how much developmental or plasticity that exists in their ability to countersing. So if countersinging itself is learned is an open question at this point. However, unlike the zebra finches and the budgerigars, their ability to produce this stereotyped song does not seem to require learning like it does for birds.
Thank you. We're a little bit over time, but a quick answer to this question. Thank you for the fascinating work. Do you plan on continuing to work with singing mouse in your own lab?
The answer is yes.
All right. Thank you. I will introduce the next speaker.